Alright, in this video we are going to talk about these mechanisms at the cellular level which are responsible for the generation and transmission of the action potential, and that for that I would be drawing this the small part of the membrane. Right, so this is the membrane which is not permeable to different ions by itself. So this is that part, that membrane part of a neuron. So we can notice that when there is no electrical current going on, so there are more sodium ions present outside. The higher concentration of sodium ion is present outside of the neuron, and higher concentration of the potassium ions are present within the neuron, right? So that gradient is higher outside, due to which the sodium tries to move in to the cell but cannot, because this membrane is not permeable. Similarly, the sodium, because of having higher gradient towards outside and more concentration inside, tries to move out but cannot go out and stays inside, and hence the resting membrane potential is restored and is maintained within the neuron cells at the time of the rest. But there are few structures which are called the voltage gated ion channels. Before I discuss the voltage gated ion channels, I would like to mention it because there is more sodium ion concentration than the potassium ion concentration inside. The outside of the membrane becomes positively charged and inside becomes the negatively charged. And this difference between these two charges is minus 70 millivolts, which is the resting membrane potential. Okay, so now come towards these these channels, ion channels. These are two types of the channels which I have drawn. One is called the voltage gated sodium channel, and the other one is the voltage gated potassium channel. And these channels basically controls the movement of the sodium and potassium ions across the membrane. Okay, so, and then the third part is the sodium potassium pump, which is a specialized protein sodium potassium pump, which uses ATP, converts it to ADP plus inorganic phosphorus to generate energy here, and that energy helps to actively transporting sodium outside and it moves potassium insides. And basically it is doing 3 sodium ions, it is moving 3 sodium ions from within the cell and moving 2 potassium ions from outside into the cell. And this process is a continuous process, and this way it will maintain a gradient, a difference between the concentration of the positive ions outside and inside of the cell, and that gradient remains always minus 70 millivolts in terms of the resting membrane potential. Right, so until now we have discussed 3 types of the structures involved in maintenance of the resting membrane potential. There are voltage gated sodium channels that regulates the movement of the sodium ions at the time of the action potential, and these gates can be opened only when there is a change in the charge, and we will see that when we discuss the action potential in the next video. The second one is the voltage gated potassium channel. Similarly, they remain closed until there is a change in the charge within the internal side of the cell membrane, and the action potential arrives here, and it results into the opening of these gates, and they allow the sodium potassium ion, and they allow the potassium ion to move out. And then thirdly is the sodium potassium pump, which continuously pumping 3 sodium ions from within the cell to the outside and 2 potassium ions from the outside to within the cell. Now I wish to mention here that these channels are known as voltage gated ion channels because they are controlled by the change in the voltage.